Hello, my name is Ashmita, and I'm currently doing a series on the coronavirus pandemic. So, before I start on with the second episode of the series, I just want to say a big thank you to all the support I've received in my previous video. I received a question in my previous video which asked me to basically explain the difference between an epidemic and a pandemic in more detail. An epidemic is a disease outbreak that occurs in a specific geographic location affecting the people who are residing in that area. So, in other words, the outbreak is contained within that region. However, a pandemic is basically a type of an epidemic, and the epidemic is basically spreading worldwide and affects a significantly large amount of the world population. The three topics that I'll be discussing in this episode are the genome sequencing of the virus, how and why scientists need to track mutations, as well as the diagnostic testing. So what exactly is genome sequencing? Genome sequencing is the process in which scientists identify the sequence of the virus in order to find the order of its four nucleotide bases. In this context, genome sequencing is needed in order to identify the origin of the virus as well as to find the unique region in order to make a diagnostic testing. Additionally, genome sequencing helps us in tracking mutations, which is essential in order for successful treatments and vaccines to be created in the future. Viruses change frequently, and most of these changes are innocuous. So, this can be thought of as a game of telephone where one person says a word to the other, and the word basically moves further and further away from its original word. So, in this context, we can use the word as the genetic sequence, which is basically the order of the four nucleotide bases. Mutations occur randomly, and scientists basically use a model to predict the history, which I'll explain later on in the video. DNA sequencing is the process of genome sequencing, and it is used to study human diseases and genetics. So, DNA sequencing is becoming more cheap, so viral sequencing is becoming more and more frequent as more viruses start to arise as time passes. The SARS-CoV-2 virus is an RNA virus, which basically means that the genetic material is RNA. So RNA is single-stranded, so it has only one strand, and it has all the four nucleotide bases except thymine is replaced by uracil. So in most organisms that have a DNA, their DNA is transcribed to RNA. So scientists use reverse transcriptase, which is an enzyme, to transform the viral RNA into its complementary DNA sequence because DNA sequencing technologies cannot decode the RNA sequence. The next topic that I'll be discussing is how scientists track mutations. So, scientists need a model of sequence evolution in order to predict the history of the virus. So, they need to track it at every location in order to help public health officials with their response to the outbreak. In this coronavirus pandemic, scientists have shared their viral genome sequencing with each other in order to build a collection to infer the evolutionary history. Such an initiative is NextTrain, which is basically an open source project that gives information to the public and to researchers about COVID 19, influenza, Ebola, and other diseases. The data from NextTrain shows us that the SARS CoV 2 virus mutates more slowly than the influenza virus. It has about 25 mutations per year. So its significantly low rate of mutations gives scientists and researchers hope that they can develop a vaccine soon. The last topic that I'll be discussing in this episode is diagnostic testing. So there are two types of diagnostic testing. The first one is RT-PCR, which stands for reverse transcriptase polymerase chain reaction. So in this testing, scientists basically take a throat or nasal swab from the suspected case in order to get the viral RNA and then transport the RNA to a laboratory where it can be transformed into DNA. Next, a specific sequence of DNA primers are added, which is why scientists need the genome sequencing of the virus first. And this process is repeated about 20 to 30 times in order to amplify the DNA. So because the RT-PCR test looks for the virus, it basically will only test positive if there is an ongoing infection. The second test is known as rapid testing kits, which is also known as serological testing. In this testing, scientists basically target the immune system of the infected against the virus. So they basically take the blood sample in order to identify antibodies that are present against the virus. Antibodies develop typically 1 to 3 weeks after contracting the virus. So this kind of testing is helpful to identify new clusters, and it also helps in detecting people who were infected but were discovered too late to be tested positive. However, since serological testing only looks for antibodies, and antibodies typically develop later in the infection, serological testing is not the most accurate way of diagnosing a person with COVID-19. Therefore, RT-PCR is a much more recommended diagnostic test. So that concludes my second episode for this series. In the third episode, I'll continue with different topics under the scientific aspect of the pandemic. As always, if you guys have any questions, comments, or thoughts, please leave them down below. Until next time, see you.